Hello! The Solis-Lemus Lab is hosted in the Wisconsin Institute for Discovery and the Department of Plant Pathology at the University of Wisconsin-Madison. Our lab uses a combination of computer science, mathematics, and statistics to transform biological data into knowledge about the world around us. In particular, we focus on DNA data or genomic sequences and develop statistical models to learn phylogenetic networks, which is the case of trees when there are hybrids, gene flow, or horizontal gene transfer. We also create synthetic data under ideal conditions where we know the true evolutionary history in order to test existing phylogenetic methods. To answer the question, can these methods estimate the correct topology? Furthermore, our lab produces software that biologists around the world can use on their own datasets. Without computer science, mathematics, or statistics, we would be limited in the amount of data that we can analyze at a time.
Now, there are two main topics that we investigate in the lab. So now we're going to get a little bit more into the details of what we do. One is phylogenomics and the other is microbiome. For the case of phylogenomics, we really study or focus on phylogenetic networks. And phylogenetic networks are this idea that it's an extension from the tree model. So we are perhaps all familiar with the idea of phylogenetic trees that represents the evolutionary relationship between different organisms. For example, in this case, the four fish. A network simply has these extra arrows that represent some form of reticulation. So it could be introgression, hybridization, horizontal gene transfer, any form of gene flow. So underlying, we have a tree structure that we know how to interpret. So we still have a root that represents the common ancestor of the clade. We have a time progression from the root to the leaves of the tree. And just if we know how to interpret trees, we know how to interpret networks because they just provide extra information in terms of the reticulation event. And you will find in literature different types of networks. The explicit type is the one that I just described, that every node has an evolutionary interpretation. So we have speciation nodes from before, just like the ones from the tree, and we have hybrid nodes that have two incoming edges as the parents. And the hybrid nodes, they represent, like I said, some reticulate event. The explicit networks also have a new numerical parameter that represents the proportion of genes that get transferred through this path. So in this case, we have a 17% estimate that the genes into this bluefish got transferred by this horizontal, well, it's not drawn horizontally, but by this green arrow. In contrast, there exist these other networks called implicit networks or split networks that they lose biological interpretation and they simply show some form of discordance in your genome or in your gene trees. The work that we do in the lab is focused on the case on the left, the explicit networks type, which we can interpret them, but they are also harder to estimate. So we need a lot more computational time. We need a lot more data. So here it is, the advertisement for the package. Phylo Networks, like I said, is a Julia package. We have a step-by-step tutorial, online documentation, and a Google user group. And this was done in collaboration with Cecile and also here at UW Madison. Everything's on GitHub, so people can just look at everything, just the code and how to use it. And we use the SNAC in the paper that we presented to estimate the network of this group of fish that had roughly 1,000 genes and 24 species of fish divided in swordtails and platyfish. And there was a lot of gene tree discordance in this group, as we can see here on the upper left part of the slide. With SNAC, we were able to estimate 2 hybridization events, one in green, one in orange. The hybridization event in green had 100% bootstrap support. The one in orange, it seemed like the target was very confidently placed at X Nezahualcóyotl, but the origin was not. It was jumping. In some of the bootstrap networks, it was up here, 75% had the origin of gene flow up here, 20% had it down here, so it was jumping around. So that covers the part of phylogenomics, and then I'm also going to very briefly talk about microbiome. that we do in the lab. And here we are interested in studying how microbial communities can serve as predictors of plant phenotypes. So we have some microbes in the soil, and we think that some characteristics in the plants can be explained by the interactions among those microbes. The interactions among the microbes are represented in what's called a microbial network here that I'm showing inside the function. And we think that these interactions, like I said, have an effect on the plant. And we want to study basically two levels. So on one side, we have the network, the microbial network, and how different forces here on the left, either from the environment or from experimental, like experimental stressors like temperature or antibiotics. We want to understand how these different forces affect the microbial network. That's on one side. And then on the other side, we want to understand how the microbial network is affecting different plant phenotypes. So that covers this very briefly the part of microbiome. To learn more about the lab, you can look at our website, or also like this QR code will take you to the lab website. And just to finish, I just want to acknowledge the sources of funding and also the students that are currently working with me. Thank you.